Hello everyone, I'm Steven and you're watching Steven Loves Science. And right now we're looking at the karyotype of a chromosomally normal male. So this male has 22 pairs of autosomal or non-sex chromosomes and 1 pair of sex chromosomes, an X and a Y, making him male, for a total of 23 pairs of chromosomes. But sometimes events can occur that cause an individual to present with an abnormal number of chromosomes. This is called numerical change, and there are 3 classes that we'll discuss as follows: polyploidy, aneuploidy, and mixoploidy. If an individual is polyploid, it means they have 1 or more extra sets of chromosomes in addition to their normal number of chromosomes. Now the most common is triploidy, and that occurs if 2 sperm cells fertilize the same egg cell. Now in humans, that would result in the immediate termination of the embryo. But if you're a plant, that results in the formation of the endosperm, which will feed the little plant embryo. So if you want that, if you're a plant, if you're a human, you most certainly don't. So we're not going to spend too much time on it, considering it does not exist in human life. So moving on to the next type of numerical change, aneuploidy. And aneuploidy occurs when you have an abnormal number of copies of 1 chromosome. So in this case, this person has 3 copies of chromosome 21. Now if you have 3 copies, it's a trisomy. So this person has 3 copies of chromosome 21, making it trisomy 21, better known as Down's syndrome. And we'll discuss this later in more detail. So how does an aneuploidy occur? Well, the most common way is an abnormal number of chromosomes is distributed to the sex cells during gamete formation. So right now we're looking at a normal diagram of meiosis. So meiosis 1, the homologous pairs are separating, and meiosis 2, the sister chromatids are separating. But sometimes an event called a non-disjunction can occur. And in a non-disjunction, either in meiosis 1, the homologous pairs will not separate properly, or meiosis 2, sister chromatids will not separate properly. Either one will result in the gametes receiving an abnormal number of chromosomes, in this case n+1. We have 1 extra, and we're missing 1 in these 2. The other way, less common, is called the anaphase lag, in which the microtubule does not attach to the kinet core properly, to the centromere, and that will subsequently cause a loss of the chromosome that will not enter the sex cell. So returning to Down syndrome, we can see that it's a trisomy of 21, and looking at this graph, we can see that the incidence of Down syndrome increases almost exponentially with a woman's age. So if a woman is past 35, she's considered to be of advanced maternal age and of a high likelihood of having a chromosomally defective child. Looking back at our diagram of non-disjunction, we've discussed a trisomy, which is when there's 1 extra chromosome in the sex cell. But what if we're missing a chromosome? That results in a monosomy. And the only survivable monosomy in humans is Turner syndrome. So Turner syndrome occurs when only the X chromosome is present. So this person will be phenotypically female, but will suffer from significant reproductive damage, will not be able to reproduce, they'll be infertile, as well as having other problems. But why is it the only survivable monosomy? Why is the X chromosome the only one you can live without? You don't need an extra X chromosome to be a woman. Well, that's because if a woman has 2 X chromosomes, a normal woman that's genotypically female has 2 X chromosomes, one of those chromosomes will become inactivated and shrivel up into something called the bar body. So here we see the bar body. So a woman is only using 1 X chromosome anyway. This just becomes heterochromatin and you're not using it. So that's why a person with Turner syndrome is able to live. And I had the picture of the calico cat here, because the calico cat is multicolored, and we can see that the X chromosome that's inactivated is randomly chosen. And in this case, for the cats, the X chromosome has the fur pigment gene on it, which is why we can see the patchiness. So you'll know a calico cat is most certainly female, except in some cases that we'll discuss on our next chromosome abnormality video. So that's Turner syndrome for you. But what about other aneuploidies of sex cells? The next one is Klinefelter syndrome. So if you have Klinefelter syndrome, it means you have 2 copies of the X chromosome and 1 copy of the male chromosome. So these individuals are phenotypically male, but they will have significant gynecomastia as well as hormone and endocrine problems. So this person, is, even with 2 X chromosomes, is able to still be male because the Y chromosome ensures maleness. The Y chromosome has a gene on it called the SRY gene, better pronounced sorry gene. I guess the discoverer was clearly a feminist. Sorry. And so that's about it for aneuploidy. So moving on to the next case we could have is mixoploidy. Now, if a person suffers from mixoploidy, it means that their condition was not congenital. In other words, they did not inherit it from their parents. They did not inherit it from the sex chromosomes being messed up. That happened during one of the first zygotic divisions. So the zygote was dividing, and the zygote had a non-disjunction. But that only affected some of the cells, but some of the cells were fine. So some zygotic cells started dividing and producing that abnormal number of chromosomes, but some of them produced normal numbers of chromosomes, and that's why we see this patchiness called mosaic. It's mosaic. It's mosaicism. Mosaic. So that can also happen in Down syndrome in the case of a trisomy 21 or in the case of any trisomy. That's why we see varying levels, we see varying degrees of the way Down syndrome affects someone. Someone can be severely affected by Down syndrome, was most likely caused by a non-disjunction in the sex cells, causing every one of their cells to be affected by the trisomy, or could have occurred during a zygotic division, causing only some of their cells to be defective. So that's it for now for chromosomal disorders, for numerical change. So please join us next week for discussing structural changes to chromosomes, structural abnormalities. And thanks a lot for watching this episode of Steven Loves Science. Please subscribe, rate, and comment.